is CORDIScovery. Hello and welcome to this episode of CORDIScovery with me, Abigail Acton. Today we're talking about malaria, from controlling mosquitoes carrying the parasite through the way malaria behaves in the body. The range of challenges facing researchers is very diverse. The UN's third Sustainable Development Goal is to, by 2030, end the epidemics of AIDS, tuberculosis and malaria. The EU is helping to achieve this ambitious goal by funding research into the disease to get a better understanding of how the parasite evolves in the body, working out why vaccines for some types of malaria remain ineffective, and how to reduce the number of infected mosquitoes in the first place. Controlling the number of infected mosquitoes, known as vectors, is still largely based on insecticide spraying. Indoor spraying and mosquito nets infused with insecticides do have a positive impact, but the World Health Organization warns that fragile gains may be lost due to insecticide resistance amongst Anopheles mosquitoes. They call for innovation to develop new tools, technologies, and approaches for vector control. Along with the difficulty of vector control, once someone is infected, the parasite Plasmodium falciparum behaves unusually within the body. It has multiple life cycle stages, and a feature of one stage may change in another. When it comes to trying to develop a vaccine, the situation is also complex. Placental malaria infected close to 13 million African women in 2022. The basic problem of placental malaria is that the molecule that the parasites used to bind in the placenta is highly variable. So an antibody recognizing one variant often doesn't bind to others, making vaccines frustratingly difficult to develop. Working in each of these fields with support from the EU research funding, today's guests are here to tell us what their work is doing to shine a light on these and other issues. Catherine Merrick is Professor of Parasitology in the Pathology Department at Cambridge University. She studies the human malaria parasite, aiming to improve our understanding of the fundamental biology of the parasite and the impact of this biology on virulence. Welcome, Catherine.
Hello, it's lovely to be here.
Lovely to have you. Lars Hviid is Professor in the Department of Immunology and Microbiology at the University of Copenhagen. He's particularly interested in improving our understanding of how people gradually acquire protection from malaria, thereby helping the development of vaccines against this deadly disease. Hello, Lars.
Hello. Fantastic to be here. Thank you.
You're welcome. Hanan Lepik is the founder and CEO of Senisio Robotics. He works at the interface of biology and mechanical and software engineering to create and deploy sterile mosquitoes on a large scale to reduce local mosquito populations. Hello, Hanan.
Hi, great being here. Thank you.
Welcome. Catherine, I'm going to turn to you first. The Plasmocycle project set out to transform our understanding of the basic biology of the malaria parasite Plasmodium and how that biology affects virulence. So can you tell us what is unusual about the malarial Plasmodium parasite, please, Catherine?
So malaria parasites really are unusual. They are single-celled organisms, and their closest ancestors are actually tiny cells, a bit like algae that live in coral reefs. So they're really very different from our own human cells. And one of the ways in which they are different is that their cell cycle, the way they make new cells, is fundamentally unusual. Most of the cells that we've studied for decades, like yeasts and human cancer cells, have basically similar biology. They divide by what we call binary fission. So they copy their genome, split it into two halves to get two genomes, and then they split the whole cell in half to get two daughter cells. And we really know a lot about those stages, the order they happen in, the proteins that control them. But malaria parasites just don't do this. They make multiple daughter cells, 20 or more, inside the same mother cell before dividing up and bursting out altogether. So most of what we know about the ordering and the control of those basic processes just can't be mapped onto these parasites.
And what did your project establish, and how did you go about establishing that? Tell us a little bit more about the work of the project.
So the first thing to say is that we took up this project because I had developed some new technology that would help us to study the malaria parasite's cell cycle. And the first innovation there was adding a gene to the parasites called thymidine kinase. This allowed us to label new DNA being made in the parasite as the genome got copied. Now, in itself, that wasn't novel. People had been doing experiments like that in human cells for years, but they just didn't work in malaria parasites. So the eureka moment here was figuring out that they didn't work simply because this one gene, thymidine kinase, was missing, and that adding it back would allow us to do all these experiments, looking at how and when the genome got copied, not in binary fission like in human cells, but in this complex syncytial cell division process in malaria parasites.
So it's almost, I mean, if I could say this, correct me if I'm wrong, but is it a little bit like analogous to the notion of a dye, where you can actually visualize something that's going on and previously you couldn't? I mean, often, like if you think about dyes being used to follow mechanisms, biological mechanisms, in this case, it was a gene that you were using to amplify and show you what was going on. Is that more or less correct?
That's right. So that we were exactly using a dye, basically, but because the parasites didn't have this gene, they ignored the dye. And if we put the gene in, now they can see the dye and we can label them with it. So we used that, just labeling the new DNA that was made on whole populations of parasites, on single parasite cells, and even on single DNA molecules in those cells to follow the basic parameters of the cell cycle.
Wow.
For example, we asked things like, how long does it take to do each new genome? How many can be replicating in a parasite at once? Because remember, a human cell is only ever doing one at a time, but a parasite can be doing a dozen at once. We even asked, how fast does a single DNA molecule get replicated? And those sound like basic questions, but they allowed us to make meaningful comparisons with future questions, like if we treat the parasites with an anti-malarial drug, do they stop making new genomes? Or if we starve them of nutrients, do they slow down? And those questions matter because they can matter for real malarial disease. Human patients can get very low on sugar, for example, and that might mess with the cell division in the parasites. Or they can take an anti-malarial drug, and we know that some of those drugs can damage DNA. So does the copying of the DNA get messed up as a result?
I think this is wonderful. It's absolutely the mechanics. And of course, you have to establish those if you're going to work out solutions. So yes, indeed. And talking about solutions, how do you feel that your work is actually going to feed into future research going forward?
Well, we are a long way off generating new interventions for malaria with this work, but the more we understand about how these parasites do cell division, the smarter we can be about designing new interventions. So, for example, to make a bad cell die, which is what we're trying to do in chemotherapy for cancer, for example, or with antiviral drugs, we often try to hit what are called cell cycle checkpoints, the way that the cell cycle is controlled. And if we can come up with combinations of anti-malarial drugs which both damage the parasite's DNA and simultaneously knacker their checkpoints, that could be a more effective therapy to make the bad malaria parasites die. And that obviously could be really impactful.
Yeah, that sounds fantastic. Absolutely super. And really well explained. Thank you so much. That was very, very clear for everyone. Thank you. Does anyone have any comments or observations to make about Catherine's work? Yes, Lars.
Yeah, well, I have one question. I mean, as I understand it, Catherine is now discussing how the asexual parasites divide inside the red blood cells. But before that stage, there is the liver stage, where one parasite actually divides not into 20-something daughter parasites, but tens of thousands of daughter parasites. Does a similar process apply in that stage? Do you know that?
It does, absolutely. And actually, some of my colleagues in Portugal are studying exactly that. And they're using the same technology that we developed, adding this thymidine kinase gene into the parasites in a mouse malaria model where you can look at that liver stage. So we can't look at a human liver very easily, but we can look at a mouse liver. And indeed, in those liver stages, thousands of new parasites are made within a single mother cell first, asynchronously, and then really interestingly, at the end, they seem to do a massive synchronous cell division, and there are then tens of thousands of daughter parasites.
Ah, fascinating. Like a great big sort of burst of them. Oh, horrible. Right. Well, I'm so glad you guys are doing this research. Lars, I'm going to turn to you now. You coordinated the PAMSEC project, and the work you and your team have done is bringing a vaccine for placental malaria potentially closer. Now, I know you're hoping to build on the work you've done so far because you feel a vaccine might be within reach. Can you tell us a little bit about what drew you into this area of research, Lars?
Well, malaria in pregnant women, what we call placental malaria, is fascinating. I mean, because it constitutes a really remarkable exception to the general rule that malaria is a childhood disease in Africa where it occurs. So adults are generally protected because they have developed immunity to the disease. The childhood malaria vaccines that are now being rolled out in Africa aims to accelerate that process. But despite this immunity, women once again become highly susceptible to malaria when they become pregnant. Without getting too technical, this is because the placenta creates a new opportunity for the parasites where they can hide and thrive. They selectively multiply there and cause inflammation, which causes placental malaria and delivery of preterm and very small babies. Remarkably, this problem goes away over successive pregnancies, which clearly suggests that there is some kind of acquisition of immunity, and that this acquisition that occurs in childhood is somehow reset by pregnancy and then replayed. And I find that fascinating.
That is fascinating. That seems very strange. Inconsistency, there must be a reason for it. Fascinating. So can you tell me a little bit more about what you think the reason might be? And in fact, actually, could you tell us a little bit more about the parasite molecule and what makes that difficult to deal with via vaccination?
Absolutely. I mean, only about 20 years ago, it became clear actually that the malaria parasites, and I'm talking like about falciparum that we're discussing here, that parasite uses a special molecule to hide in the placenta. And once the immune system sees that molecule, it starts making an immune response to it. It produces antibodies that can prevent the parasites from sticking in the placenta. So the next time the woman gets pregnant, those antibodies are there for her, and the problem of placenta malaria is no longer her problem. The molecule that the parasites utilize, we call it VAR2-CSA, is of no use to parasites if there is no placenta to hide in. Women, therefore, do not start to make antibodies to this molecule until VAR2-CSA positive parasites appear during pregnancy. And once the woman has acquired these antibodies, she's protected. So placental malaria is therefore mainly a problem during the first few pregnancies, because afterwards she has acquired immunity.
And if we know what the antibody is, how come we can't harness the power of a vaccine to boost immunity in the first place?
The discovery of the VAR2-CSA molecule spawned the idea that it should be possible to make a vaccine specifically to give to young women prior to their first pregnancy, and then to the aim to protect them against placental malaria. VAR2-CSA vaccines have been tried, and they should work, but they haven't really given very encouraging results. VAR2-CSA is a tricky molecule. I mean, even though every parasite has at least one, they are not completely identical between different parasites. But to be useful in a vaccine, the vaccine needs to target the exact part of such a molecule that is conserved among all the variants. And this was not achieved in the vaccines that have been tested. And therefore, they could only protect against those variants that were used in the vaccine.
So it's a question of looking for a common denominator.
Yes.
That could be targeted by a vaccine.
Exactly.
Okay, I see. So what did the project set out to do and how? Tell us a little bit about the technology that you were using, because I do understand that drove really back the frontiers.
Yes, I mean, we were struck actually with this result of the vaccination, which disappointed us as much as anybody, really, was that in contrast to this marked variant specificity, the antibodies that women acquire in response to placental malaria, they seem to be quite cross-reactive. And it is actually this discrepancy between the variant specificity after vaccination and the apparent cross-reactivity after exposure.
Cross-reactivity?
Cross-reactivity meaning that the antibody can recognize many different variants of this VAR2-CSA molecule.
Okay, fine. After exposure?
So we wanted to set out to find out why that was. The evidence that it's cross-reactive after natural exposure was quite flimsy, actually. We only had a handful of antibodies that we could use to show that. But those were antibodies that we discovered some 20 years ago. But Pilar Quintana, who is a very smart scientist working with me, she proposed that we should use a new technology that was called LibraSec to dramatically crank up the number of antibodies that we could study. So what Pilar did was that she went to the lab in the US that had developed this technology, and she applied it, but using different variants of our VAR2-CSA molecule.
Right. And what did you find?
Well, she managed during her period over there to identify about 1,000 new antibodies. I mean, which was quite a leap from the initial handful that we had. And even better, those antibodies most of them were quite cross-reactive. So the tentative conclusion that we made many years ago that was based on a handful of antibodies seems to be holding true now that we have tested it with many, many more.
Oh, that's wonderful. That's really, really excellent. And Lars, I know that the work you've been doing is really fundamental, and as you say, really broadening our ability to analyze very many more of these antibodies. What are you hoping to be able to achieve in your next stage of research if you get the funding?
Yeah, well, we need to find out exactly why it is that those antibodies that are naturally acquired are cross-reactive, whereas the vaccine-induced antibodies are not. We actually think we know why that is, but we, of course, need to be sure. So if you consider the VAR2-CSA molecule as a piece of string or piece of rope, when you do a vaccine, in practice, you take a short piece of that string and you immunize with that. So the antibodies you generate can only recognize something on that little piece. But in reality, this molecule is not a piece of string. It is folded up like a knot. So what we think is that the important things recognized by antibodies are composed of little bits on the string that when the string is straight, they are far apart from each other. But as you knot it up, then they will come close together. So when you use a vaccine, you break these things that are recognized by the important antibodies, and therefore the vaccine is not cross-reactive. We are now testing this. I mean, if it's correct, we then need to find out how can we make the molecule look more knot-like.
Right.
But we hope that this work will somehow make the problem of malaria and placental malaria go away one happy day.
Well, that would be just obviously amazingly important, and the work you're doing is so groundbreaking. So I really hope that you can build on it and continue to build on it, because I think it's clearly forging a way towards a vaccine for this horrible condition. So thank you very much, Lars. Do we have any questions for Lars? Hanan, yes, I think you have.
If a very naive question, if I may.
I love those. They're excellent. It means that I don't have to ask them.
I fear those.
So it's amazing that you said that at subsequent pregnancies of women, they have lower chances on contracting malaria, right? Is there a chance somehow to maybe trick the parasite and let them think that, like, she will take a pill or something, that her body is now pregnant while it's not, and then by that lowering the chance it will contract the malaria?
Well, that's a good question. I think actually the parasites, you don't need to try to trick the parasites, because I think that the parasites are always trying to figure out whether their host is pregnant. So they are continuously trying to express this VAR2-CSA molecule. But if they happen to be in a non-pregnant individual, it could be a non-pregnant woman, it could be a child, it could be a man, those parasites will die. And therefore, you don't make an immune response to it. So people like you and me will never make antibodies to that, even if we get malaria.
Yeah. Okay, super. Thanks very much, Lars. And I really hope that your work can continue. It's certainly very, very significant work. Yeah. Hanan, I'm going to turn to you. We've been looking at the biology. We've been looking at the notion of identifying and perhaps finding a solution. But if we go right back down to basics, what we really need to do is control the number of mosquitoes. So the RoboCit project has used AI and other tools to successfully breed, which isn't easy, and disperse, also not easy, sterile male mosquitoes as a way to control vector populations. So can you tell us a little bit more about the current ways, Hanan, now, prior to your innovation? What are the ways that are currently used to control numbers of mosquitoes, please?
Sure. So typically, specifically also in Africa, there was a huge campaign delivering and providing bed nets, which did a great and effective work, pesticides. So that's great that used to be great for adults. And the thing is that once the fogging is done and evaporates, then you are still left with all the eggs and the larvae and the pupa, which are still currently developing, other than the fact that it's typically unhealthy. Also, on top of that, pesticides, they developed resistance to pesticides, and creating new materials is a very tedious and long regulatory process. We saw the success of the sterile fruit flies program. So, for example, in Guatemala, where one of the largest SIT sterile insect technique facilities in the world resides, they produce about 1 billion of sterile fruit flies every week or so. I believe this is like the gold standard for mosquito control. Specifically for the Anopheles, for the malaria mosquito, there are huge challenges in the ability to separate the females from the males. It's typically much, much easier at the adult stage. So we want to separate the males from the females, and we can do that at the Anopheles at the adult stage. The mosquitoes are very fragile, and we want to rear millions of them and then release them into nature, streets, wherever.
Right. Okay. So that's the target. That's the ideal. What is Robosit doing?
We took this concept and identified why it was not used so far on global scale. You need a lot of working hands. You need a lot of people in order to rear the mosquitoes. You want to release males only because the females bite. And then the males are being released. They target the females. They mate. There's no baby mosquito. And as the female mosquito typically mates only once in a lifetime, when you're releasing once a week, then the population drops within X number of weeks or months, depending on the infestation level. So you need a lot of people, and you need to be repetitive. So what we did is we are harnessing AI and automation. When you're looking at an adult mosquito, it's very easy to understand whether it's a male or a female. Very easy. I'll show you one picture. You'll be an expert in no time. And I realized that when they are emerging, then they need to, they're standing in order to dry up the exoskeleton coming out of the water, and are standing there. While they are standing, they're immobile. When they do that, you easily identify whether it's a male or female. So it hit me that we need to find a way to immobilize mosquitoes, to identify them, to use vision to identify whether it's a male or female.
So what did you do next? So once they're identified, what's your process? How do you separate the males from the female?
So long story short, we have built an automated factory made of 3 components. One is the emergence or final rearing stages of the mosquitoes. They are inside the water, and they are being guided through air ducts towards the QA sorting imaging station. When they fly and being guided through the air duct, they land on a conveyor belt, which is porous. And the air acts as a pull force. So the mosquitoes are standing on the conveyor belt without the ability to fly away. They may be able to walk a little bit, they are not able to fly away. So imagine a factory which package cookies, and you have a conveyor belt with millions of cookies all the time on a conveyor belt. It arrives towards an imaging station. The computer identifies the cookies on the conveyor belt, and something takes out the cookies and put them into boxes and then ship them to the supermarkets. So pretty much the same. So now we are able to source with the emergency units millions of mosquitoes without touching them. We identify in real time in a capacity of more than 300 mosquitoes per second. I'll say it again, more than 300 mosquitoes per second.
And that can identify the males from the females.
Yeah, this is unparalleled to any capability that any human can do or any other manual process currently on Earth.
And then what happens to the males?
So we take out the females, and then conveyor propagate, and we load the males into release canisters. We then take them and irradiate them.
And that makes them sterile. So it's the process of irradiation that makes them sterile?
Right. Correct. And then we provide them to the distributor, the pest control company or whatever, and they go to the field and they can release them. So we can cover very large areas from a single facility, from a single factory. And by introducing this process, the So the entire process is industrialized. Now, at the end of the week, we change the software mode, and we then load also females, and we take them back to the first room. So now we have both males and females for the next week. So it's cyclic, okay?
Okay, so you're kind of recycling the females, essentially.
Yes, exactly.
I understand that you were speaking to some people at the COP with regard to the using your sterile males in Africa. Can you briefly tell me a little bit more about that?
Yeah, so we presented Israel, part of the Israel technology delegation, the latest COP29 in Baku, Azerbaijan. And there it was really an amazing experience. We met people from across the world, different colors, customs, cultures, all of them interested in climate. So malaria is not only in Africa, and mosquitoes, as carry different, are responsible for different diseases. The malaria, the chikungunya, dengue, and others. So there we met a bunch of the different representatives from Ghana, from Uganda, from Burkina Faso, from Tanzania. They were excited and thrilled about this potential because they have all those problems with the malaria, and some of them already heard about the SIT, but they have problems of scaling up. So seeing a true solution, leveraging, harnessing the power of AI to do something good and be able to scale up this technology and this solution is very exciting to all of them. And we're already now discussing with some of them, also together with the representatives from the UN who are at the COP and now helping us. So I really see really exciting potential to roll this technology in Africa.
Very good. Excellent. Thank you so much, Hanan, for the explanation. That was really, really interesting. Well, this is just wonderful. Thanks very, very much for your time explaining your work, which is very interesting. And I'm very glad that we've tackled this subject from various angles. I think that's gives us a good overview. So thanks for your time and for joining me today on CORDIScovery.
Thank you very much.
Thank you very much for having me.
Thank you.
You're very welcome, everybody. Carry on with the good work. That's all I can say. Okay.
Bye-bye.
Goodbye. Bye-bye.
Bye.
If you've enjoyed this podcast, follow us on Spotify and Apple Podcasts, or whichever service you use, and check out the podcast homepage on the CORDIS website. Subscribe to make sure the hottest research in EU-funded science isn't passing you by. And if you're enjoying listening, why not spread the word? We've talked about training bees to fly through hoops. Listen to the episode on hidden interactions to find out why. In our last 42 episodes, there'll be something there to tweak your curiosity. Perhaps you want to know what other EU-funded projects are doing to reduce the number of deaths from malaria. The CORDIS website will give you an insight into the results of projects funded by Horizon 2020 and Horizon Europe that are working in this area. The website has articles and interviews that explore the results of research being conducted in a very broad range of domains and subjects, from neutrons to neurons. There is something there for you. Maybe you're involved in a project or would like to apply for funding. Take a look at what others are doing in your domain. So come and check out the research that's revealing what makes our world tick. We're always happy to hear from you. Drop us a line, editorial@cordis.europa.eu. Until next time.